Good evening to all in Korea, and good morning to Professor Langer. My name is Hyukjin Lee from Ewha Women's University, and it is my pleasure to welcome you all to this event with Professor Langer. This is the second online talk series with an eminent scholar, hosted by the Korean Academy of Science and Technology. It is also my honor to have Professor Yu-kyoung from Seoul National University. She is the Dean of College of Pharmacy at Seoul National University. Before we get started, I'd like to welcome Professor Min-Koo, the President of KAST, to give us welcome remark.
Good evening, everyone, and good morning to Professor Langer. First of all, I would like to express my sincere gratitude to Professor Robert Langer of MIT, Professor Yu-kyoung of Seoul National University, Professor Hyukjin Lee, Ewha Women's University, and all of the audience who are joining the program of online talk with Professor Langer through YouTube. We all know that Professor Langer may be the most outstanding professor in our times. Speaking of his academic achievement, such as controlled release system and the transdermal delivery system, etc., we were able to reaffirm its significance through this pandemic. Professor Langer is also a successful entrepreneur, having initiated more 40 biotech companies, including Moderna. Today, I'm very much looking forward to seeing how the conversation will unfold and motivate students, researchers from both scientific and the industrial field. Last but not least, I believe that this might be once in a lifetime chance for the audience. I would like you to actively participate in the dialogue with Professor Langer and hope today's program will be a memorable time for all of you. Thank you.
Professor Han, thank you for the warm opening remark. Now it is my pleasure to introduce today's speaker. Professor Robert Langer is the Institute Professor at MIT, and he published over 1,500 articles, which have been cited more than 350,000 times. His H-index 294 is the highest of any engineer in the history. His patents have been licensed or sub-licensed to over 400 different companies. He is a co-founder of Moderna. Today, he is joining us from United States. Good morning, Professor Langer. Can you hear me?
Yes, I can. Nice to see you. Thank you.
Great to see you again. So, where are you joining us from now?
I'm actually at my home in Newton.
Okay, great.
So I go into MIT about half the time, but a lot of times I have to do Zoom things. So
it's always good to see you in good health. Professor Langer will give us a presentation about his research and achievement. And Professor Langer, can you begin whenever you are ready?
Yes. Thank you so much. It's a pleasure for me to be able to speak. So I thought I'd tell you about some of the things we've been doing in drug delivery and how I got started in it, which was kind of an unusual way. If we could have the slides on, please. Can we have the first slide? Thank you. So anyhow, the way I got started, I'm actually a chemical engineer, and I got my degree from MIT in 1974. And when I did, pretty much all my friends went to work in oil companies. So I thought that's what I would do too. And so they had enormous number of openings. So I applied to oil companies. I actually got 20 job offers, 4 from Exxon alone. And I wasn't that good, but they just had so many openings. But when I went there, I wasn't very excited about it. They'd say, well, gee, if you could just increase the yield of this one chemical by 0.1%, they said that'd be worth billions of dollars. And I remember flying back to Boston, thinking I didn't want to do that. So then I started thinking about other things I might do. And one time I saw an ad to the assistant professor at a university, City College in New York, to create chemistry curricula. And I really was excited about that because when I was supposed to be doing my PhD thesis at MIT, I had spent a lot of time starting a high school for disadvantaged high school students. And I got very involved in creating new chemistry curricula. So anyhow, I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea. So I found all the ads I could to be assistant professor to develop new chemistry curriculum. I found about 40 and I wrote them all, and actually none of them wrote me back. I guess I wasn't in the right box, you know, so that was unfortunate. So I didn't get any of those jobs. And then I started thinking, what other way could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools too, and they didn't write me back either. But then one day I walked into the lab where I was, and one of the older scientists said to me, Bob, you know, there's a surgeon in Boston named Judah Folkman, and sometimes he hires unusual people. He actually thought very highly of Dr. Folkman. I won't say what he thought about me. Anyhow, if we could go to the next slide. This is the idea that Dr. Folkman had in the early 70s about how tumors grow in relation to their blood vessels. And if you look in the bottom left, his idea was that the tumor cell would somehow become abnormal. It would then, if you follow the arrows, grow in like a three-dimensional mass. And if you go to the middle, bottom middle, maybe it has 1 million cells. And actually, when it gets to that part in the bottom middle, he said it wouldn't be able to get bigger than that because it runs into a nutrition problem. Cells in the center die because they can't get nutrients or get rid of waste. But what he said is that what the tumor does, which is very unusual, is it secretes what he called a chemical signal called TAF, or tumor angiogenesis factor. And what that does is it diffuses to the surrounding blood vessels, which are normally quiescent, don't do anything, and it causes them to grow right to the tumor. And when that happens, then that solves the nutrition problem for the tumor. It gets bigger and bigger, and you see this on the upper right-hand panel. And of course, when it gets very vascularized, not only is it large, relatively speaking, but it can spread through those blood vessels. That's a process called metastasis, and that's often what kills people. So his idea was that if you could stop the blood vessels, in other words, achieve anti-angiogenesis, maybe that would be a whole new way of thinking about stopping cancer. Now, I thought this was a terrific idea, but I didn't know any biology. And it turns out everybody criticized him. They thought he was wrong. But he hired me, and my job, he said, was to prove he was right. And in so doing, to isolate the very first substances that would stop blood vessels. Now, one of the key things whenever you're trying to do a project like this, you know, discover new bioactive factors, is you need what's called a bioassay. And there was no bioassay to study blood vessels, so we had to invent one. And what we did is shown on the next slide. We published this in Science, but our idea was that certain types of tumors you could put in the eye of a rabbit, and they would cause blood vessels to grow in the eye, and you could actually see them because the eye, unlike any other part of the body, doesn't have background blood vessels. The cornea doesn't. So, but it takes maybe 8 weeks for this to happen, as I'll show you. So the idea is the tumor secretes the signal, and then blood vessels will grow. But the molecules we were trying to isolate to stop blood vessel growth, they were pretty large molecules. So we needed something like a polymer or lipid that you could put in the eye that could deliver these large molecules for a reasonable amount of time. Also, they shouldn't cause inflammation in the eye. So this was very challenging. And Dr. Folkman was on the board of the one company in the world, ALZA, that had done any work in this field. And they had Nobel Prize winners and other great scientists on their board. And he asked them, could they help? But they said, no, this is impossible. The next slide shows basically what they said, that these large molecules that can't slowly diffuse through solid materials any more than any of us could walk through a brick wall. They just said they're too big. And the literature, if we go to the next slide, said similar things, that the use of these matrices is virtually restricted to small molecules. But if we were going to solve this problem of angiogenesis, we had to come up with a way to do this. We had to find a delivery system. So I spent several years experimenting with different techniques. And I actually found over 200 different ways to get this to not work. But eventually I found a way to get it to work. And I was able to make little microspheres or nanospheres. These are shown on the next slide. And you see a picture of one on the left and cut in half on the right. And what we were able to show when we published in the journal Nature in 1976, next slide, was that you could use this approach to deliver really molecules of any size, almost for any length of time. And in this Nature paper, this is just one graph from it. We delivered these different molecules for over 100 days. Also, this paper, to my knowledge, is the very first time that anybody had ever made tiny particles and delivered nucleic acids, like nucleic acids are DNA and RNA. And of course, as I get to Moderna later, that will become very important, as people know, because the delivery of nucleic acids is complicated, and people didn't think you could do that. At any rate, when I first presented this work at a scientific meeting, I was 27 years old, actually, at the time. It was a long time ago. I'm 73 now. But when I remember presenting this at a big scientific meeting with many famous material scientists there, I thought it's a 20-minute talk. I thought I did okay. I didn't forget too much what I was going to say. So I thought when I was done with this talk that these older scientists would want to encourage me, this young guy. But when I got off the podium, a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said, well, the next slide shows some of what they said, that large molecules can't slowly diffuse through the solid materials. That's what I mentioned before. And they also pointed out that the organic solvents we're using, like alcohol and acetones and other methylene chlorides, that they would destroy any of these large molecules, peptides, proteins, or nucleic acids that we would be putting in the nanospheres or microspheres. And actually things went downhill from there. I applied for grants and my first 9 grants were rejected. The reviewer said, well, this bio stuff that I'm doing doesn't make any sense for a chemical engineer to do. This, like I said, was the 1970s. And then I applied for faculty positions, and my first 9 and my first every single faculty department in the world in chemical engineering, that's my discipline, turned me down. So I couldn't get a job. But then Dr. Folkman knew Nevin Scrimshaw, who was a very famous nutritionist, and also on that ALSA board, and he introduced me to him, and Dr. Scrimshaw offered me a job. But Dr. Scrimshaw was kind of a benevolent dictator kind of department head, and he didn't ask anybody else what they thought. So, and that would have been okay, except for the year after I joined the department, Dr. Scrimshaw left. And so a lot of the faculty decided to give me advice, and their advice is I should leave too. In fact, the next slide, this is a direct quote from Michael Marletta, a good friend of mine. He was giving a speech a few years ago, and he was talking about when he was at MIT with me. And this refers back to 1979, 1980. And he said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." So that was very discouraging, but I kept trying. And we had made progress on the drug delivery system, so we were able to do the bioassay. And the next slide is just a repeat of what that looks like. And then what we did, and if people don't like the sight of blood, you shouldn't look at the next slide, but there's no other way to illustrate this, is that when we tested maybe 100 different fractions in literally 2,000 eyes. And when you didn't have an inhibitor, you see what you see on the left, a sheet of blood vessels growing over the polymer towards the tumor. And this happened every single time, 1,000 out of 1,000, 2,000 out of 2,000. And if you looked at these eyes just 3 weeks later, it would be three-dimensional, the tumors. Out of the orbit of the eye would actually kill. But when you put the inhibitor in, it's striking. We see on the right-hand side that at the same time, the vessels are sparser, they're lower, they avoid the polymer in the tumor. And 60% of the time, these tumors never grew, which is just shocking because 100% of the time, and on the left-hand side, they did. And we repeated this experiment on the right 21 times. So that showed for the first time, we published this in Science in 1976, that angiogenesis inhibitors existed and provided techniques by which everyone else could study them in the future. Now, this was 1976. It took another 28 years before the first angiogenesis inhibitor was approved by FDA. And let me just show you what happened starting in 2004 on the next slide. One after the next inhibitor would be approved by FDA. Avastin maybe is the number 2 or 3 best-selling biotech drug in history. And not only are these drugs used for treating cancer, all kinds of cancer, they're by millions and millions of people, but they're also used for treating eye diseases that heretofore there was no pharmacologic treatment. So now today there are drugs like Lucentis and Eylea that can stop blood vessels from growing in the back of the eye to actually, in some cases, reverse and treat diseases like macular degeneration and diabetic retinopathy. So another thing that Dr. Folkman and I talked about, which might sound common today, is filing a patent. You know, at 1970s, Boston Children's Hospital, you know, one of the best hospitals in the world, they never had a patent. And Dr. Folkman said, well, we should file for one. So we did. But 5 years in a row, the patent examiner rejected it. So the head of the tech transfer office at the hospital came to see me one day, said, Bob, you are just wasting so much money on these patents, you know, and the examiner's never going to allow it. You should just give up. But you can probably tell by now I don't like to give up. So I started to think, how could we convince the examiner and to allow it legally, of course. And when I mentioned in the beginning, when we tried to do this, lots of people told us it was impossible. It would never work. I wondered, maybe somebody wrote that down. So I did what's called the science citation search. I did it in 1982, 6 years after the Nature paper, to see who wrote about our work. And let me just show you one paper that I found. I had no idea it was written, but it's on the next slide. And what these people wrote, and they were 5 of the top material scientists in the world, they're describing the field in 1979. And they said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so surprising is a really good word for a patent examiner, that clearly demonstrate the opposite. So I showed this to the person at the hospital. He flew down to Washington to show it to the examiner. And the examiner said, gee, he had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 people that they really wrote that. So I wrote them, and they were all nice enough to write me back that they really wrote it. And then we got this very broad patent. Next slide. And with that, we taught different companies how to do this. I got involved in starting companies, as was mentioned in the introduction. And today, if we go to the next slide, there are many, many products based on this. And they're absolutely critical to have these, because if you didn't put them in microspheres or other things, they would get destroyed. Now, we wanted to take this further. I mentioned that we could make nanospheres as well. But of course, when you make nanospheres and you inject them into the bloodstream or the body, a lot of times they don't last very long. I mean, they could release for a long time, but they could get eaten by macrophages. So we designed a certain type of coating, polyethylene glycol, to prevent that from happening. And I thought I'd just show you a video of this. Next slide.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should let you know it doesn't really blow the cell up like that. But the beauty of this then is that now you could deliver small molecules, or, as I'll get to, siRNA or messenger RNA. And that brings me to Moderna. In 2010, if we could go to the next slide, Derek Rossi, Ken Chen, Nubar Afey and myself started Moderna. And the idea is sort of shown here, that DNA makes RNA makes protein, right? That's a central dogma. And of course, protein therapeutics had been hugely successful by then. In fact, most of the best-selling drugs right now are protein drugs. But protein drugs are hard to make. I mean, it takes years sometimes to make them and purify them, and also there's a lot of diseases you can't treat with protein drugs, like intracellular diseases, membrane-bound diseases. The beauty of messenger RNA is it can solve all these problems. If you could deliver messenger RNA to the body would do all the work and make the protein and hopefully treat those diseases. Of course, you need to protect the messenger RNA. And again, that's where the nanoparticles come in. So with this idea in mind, we started Moderna. Moderna, by early 2000, had 13 products in clinical trials, all in nanoparticles with polyethylene glycol on the outside. Then the COVID crisis came. And to show you the power of messenger RNA compared to any other technology, let me just go to the next slide, which talks about the Moderna COVID timeline, which was pretty unprecedented in terms of how fast this moved. On January 11th, the Chinese scientists published the virus genetic sequence. It took just 2 days for Moderna to then finalize the messenger RNA design from that. Within another month, pretty much, they'd made the vaccine and the nanoparticles, shipped it to NIH for testing, and within another month, less than a month, the first clinical trials had started. And of course, as you all know, by the end of last year, it would get approved for emergency use authorization. By the way, the other company that's done this, Pfizer and BioNTech, I mean, they're also putting it in nanoparticles. Nonetheless, what's happened when we started doing this, now today it seems like, of course, it would work. But you know, just like I talked about my own career, when we did Moderna and did the work, people really criticized it. People kept saying, this doesn't make any scientific sense. Just let me just show you on the next slide a picture from the Boston Globe, our local newspaper, and the front page headlines, you know, just a little more than a year ago. And they have my picture on the cover, and they had talked to a lot of scientists and clinicians, and the headlines is, this is not how you do science, which is pretty discouraging. But of course, all of them were wrong. And as you know, next slide, that the Moderna vaccine is now used all over the world. And so is too is the Pfizer BioNTech, all, like I say, using nanoparticles with polyethylene glycol on them. So, and messenger RNA inside. So what we also started thinking about over the years is, can we not only deliver things by injection, but could we even deliver drugs orally? You might think, how could you deliver complex drugs like RNA or proteins orally? Well, if we could go to the next slide, you know, what are the three issues that prevent delivering anything orally? Well, first, it's going through the stomach, and there's acid. Then there's degradation from proteases. And finally, you have to diffuse through the stomach or intestinal wall, which is a tough barrier. And so those are the three problems. Now, one way to solve all of them is to inject something into the stomach. And in fact, that's been done, which is, I'm going to show you on the next slide. People inject things into the stomach all the time, but they're always going through your rear end, right? I mean, so you do what's called an endoscopy, and you can deliver injections into the stomach. It's actually very highly efficient, doesn't really cause much pain because there's few nerves there, and the tissue will regenerate rapidly. But of course, I don't think anybody in this audience, you know, wants to do injections through your rear end. But could you ever make a pill that could do the injection? So here's what Gio Traverso and Alex Abramson and myself did. We said, well, could we make a pill? And the pill, it's actually very tiny. You know, you put it in a capsule. But basically, the idea of this pill is that there's a little hole in the surface, but then what's behind that hole is a needle. Now, the needle can't come out unless we push it out. The needle, by the way, is made of almost entirely the protein, like insulin, which is the model that I want to talk about, but you could do this for anything, and we have. So you have this needle behind the little hole in our pill. Behind the needle is a spring, and the spring is cocked, and the spring will release the needle, automatically if you want it to. But of course, you don't want it to be released right away, right when you're in the mouth. You want it to be released maybe 30 minutes, an hour later when you're in the stomach. So what we've done is we've embedded the spring in what's called a sugar glass. So it can't break until maybe 30 minutes or an hour later. And you can control the time of that by water coming in. What causes the sugar glass to break is osmosis. Water comes into the pill, and then it causes the sugar glass to break. That releases the spring, and then the spring shoots the needle out this little hole. There's one more problem though. How do we know that that hole is always going to be facing the stomach wall? So what we did is we modeled our little pill after a leopard tortoise, which has an interesting weight distribution, so that when it tumbles in the stomach, it will always tumble, if we could go to the next slide, such that the hole is facing the stomach wall, and then the needle just shoots out. So the device localizes to the wall, it self-orients itself, and you can give a dose that's clinically relevant. Let me just show you this leopard tortoise is what we modeled it, and on the next slide. Like I say, it always will do this. The tortoise is on the left, the needle on our pill on the right, but it always does this, and it will do this 100 times out of 100. We published this in Science. I'll also show you the spring, and the idea on the next slide is that when you release the spring, it just shoots the needle right out. But the spring itself can't get out of the pill, but because it's too big. But the needle, which is made out of all insulin, gets out, and you can control the time that it comes out simply by changing the diameter of the sugar. And our insulin, just to show you the perforation, if we go to the next slide, the perforation risk is really mitigated by controlling the force. And if you look at histology, it's actually, like on the right, it's actually as good as conventional systems like Carlock needles. Just to show you a few other things, it's actually, the insulin's quite stable. We see this on the next slide. And you can use this approach for other things rather than just insulin. The next slide just shows some examples of lysozyme and glucose 6-phosphate dehydrogenase. Finally, does it work? Well, we're in animal trials now. We have a partner with which is Novo Nordisk, and they're going to start human trials soon. But let me just show you from our Science paper whether it works in pigs, which is a pretty good animal model. And the next slide, what we're looking at is insulin in plasma insulin with this approach. And whether you give it by pill or by sub-Q injection, you get pretty much the same result. And if you look on the right-hand side, you see histology, and really there's no tissue damage. And so that's looking at insulin concentration. But of course, if it works, you should also lower blood sugar levels. And the next slide shows that you do. And in fact, you lower it pretty much the same as the sub-Q injections. And again, when you look at the histology on the right-hand side, there's really no tissue damage. So I thought I'd try to go over one last thing, on how we might deliver things. And now I want to talk about delivering cells rather than just drugs. And here Jay Vacanti and I have collaborated literally for over 40 years on trying to create new tissues and organs. Let me just motivate it by showing you a picture of a little baby. And he's dying of liver failure. So the only way he'd live is somebody else dies and you do a transplant. But Jay and I had an idea, and that idea is shown in the next slide. This is from a paper we wrote in Science, the journal Science. It's been cited over 8,000 times, and it goes over our strategy that you would take different cell types. If you injected them at random, nothing happens, but the cells are smart. And if you put the cells close enough together on a scaffold, they would form structures, and you grow them in bioreactors, and then you could put them in a person. Now, we also, when we did this, we didn't even talk about stem cells. I mean, nobody talked about stem cells because they weren't really being used, but now you could use that strategy, and as we do, with stem cells as well. Anyhow, just to show you a few pictures, you could make a scaffold. If we go to the next slide, you could have basically scaffolds with cells, and you can make these in any shape. In fact, one of the things that some of the Vacantes did is make them, and Linda Griffith, who was one of my former postdocs and now professor at MIT, is you could make ears. In fact, the Army approached us to see if we could even make scaffolds in the form of ears for soldiers who returned from Iraq and Afghanistan. And the next slide just shows a picture of that. On the left is the picture of the ear, and on the right, the scaffold, where you have cartilage cells making matrix. Now, these are actually used clinically for helping burn victims and patients with diabetic skin ulcers. I'll just show you an example of that. We licensed our patents to different companies. And the next slide shows a baby who's very badly burned, but he's 2 years old. But you can take the scaffold, which you could cryopreserve, which has neonatal skin fibroblasts on it, and you could put it on the child at the time of the injury. Next slide. Now, if we go to the next slide, we're putting it on the child. That's time 0. Let's come back 3 weeks later. Next slide. He's doing better. Let's come back 6 months later, and next slide. Yeah, he's pretty much healed. And these are now approved by regulatory authorities for patients with diabetic skin ulcers and burns. And there are many other groups working on making new tissues and organs. Laura Nicholson has a company. She was one of my former postdocs, called Humacyte, that's making new blood vessels. There are other companies making spinal cords for people who are paralyzed. Companies like Frequency Therapeutics trying to address hearing loss, making new hair cells. Other companies like Sigilon making new pancreases, and on and on. But not only can you make new tissues and organs, hopefully for patients, but this type of technology may also reduce animal and human testing someday, because you can make organs and tissues on a chip. And to that end, Gordana Vunyak, who is one of our fellows, and Milika Ritzek, who's now at the University of Toronto, one of our graduate students, actually created a heart on a chip. And just to show you a picture of that, they took, on the next slide, they took heart muscle, and when you do, you get the characteristic structures of sarcomers and intercalated discs. And what I'm going to now show you on the next video is a video of putting 6 million cells on one of these scaffolds. If we could go to that video. And when you look at this, you might think we're shaking it, but we're not. It's actually 6 million cells beating as one, just like a little heart. We'll just watch that for a second or two. And so it's really, like I say, just like the heart, and you can do drug testing. You can even do an EKG. The next slide shows you the EKG. That's actually the last slide. And so you could actually use this to, and they have a little company called Tara Biosystems, and they're working with many large companies to screen their drugs for safety and efficacy, which could reduce animal testing and speed up and accelerate discovery. And others are also developing technologies on a chip. Gio Traverso, who I mentioned, is now at MIT and Harvard, created a gastrointestinal tract on a chip, which can tremendously accelerate drug delivery. Before this, people only use CACO2 cells, but they're not very accurate. And he has published with us a paper in Nature Biomedical Engineering, and there's a little company, Vivitex, that can do rapid testing and use these systems to figure out excipients that can get all kinds of molecules that normally you couldn't get absorbed. Now they can be. And others, we're also working on brain on a chip with Li Wei Sai at MIT. So all these things, I think, could someday happen. And then there's even people who are making meat using these approaches, or making leather. So anyhow, that's what I want to go over today. It's really an honor to speak with you, and I'm happy to answer any questions. Thank you so much.
Great. Thank you so much for your inspiring talk. We have received a lot of questions from our audience. Now,
Sure.
Professor Yu-kyoung will ask these questions to Professor Langer.
Sure, certainly.
Good morning, Dr. Langer. It's a really super impressive talk, as always. And we got many questions from the audiences in advance. And as you may know, now Moderna vaccine is pretty much popular in Korea. So even the kids in elementary school know about Moderna. So, so if you don't mind, can I ask some questions about the Moderna?
Anything you want.
All right, okay. Then it's a relief. So it is really amazing that the mRNA vaccine came through and it really saved all the world. But behind the brilliant success story of the Corona Moderna vaccine, I think you may have gone through some challenging part. So when you developed the Moderna vaccine from bench to bed, what was the most challenging part? Can you share with us? I mean, if it's not that much confidential.
No, I don't think it is. Well, I think it really depends how you look at it. You know, if you ask our CEO, Stefan Bancel, what he wishes he did differently, of course, I think he's done unbelievable, but he always says that, you know, he wished he even, you know, Moderna made a giant multi-million-dollar investment in a manufacturing facility near Boston in Norwood, and it was, I think, a huge risk for him to do that, but he did. And, you know, we as a board approved it, and I think it was great. But he would say that he wished he spent even more money on developing, on the manufacturing, because so many doses are needed. But I think that probably would be the one thing he'd say, because we wish that, you know, we could have made double the doses or triple the doses. We're doing that now. We're expanding the manufacturing facilities, but you know, you can't do that overnight. But that's what he has said, and I would agree.
All right, so I think for that kind of large-scale manufacturing, I think maybe Moderna have used the microfluidic technology. Is it right? Microfluidic, or if
Initially, they used microfluidics. They actually keep improving the technology.
I see. So do you think that kind of microfluidic technology, that kind of concept, do you think it is really essential for making the large amount of mRNA vaccine? What kind of modification do we have to do to make that kind of large-scale mRNA vaccine? Do you have any suggestions?
Well, I think that's a great question. You know, of course, we started Rohit Karnik in our lab many years ago, and Omid Farrokhzad did some of the first microfluidic manufacturing of nanoparticles. And so I certainly think that's one way to do it. But I think that, and I don't want to be too specific, because I think there's probably variety of strategies that people might use. But you have to make sure then you get the same quality control that you have with what we're doing now, and you have the ways to characterize the nanoparticles and the messenger RNA. But I don't know that one's limited to that. You have to, the key issue, just to go over it, is you have the messenger RNA, and generally there's at least 4 lipid components that you have to mix together in a certain way. And so that you have to make sure that they then surround and protect the messenger RNA. And like I say, there's the 4 components, and so it's non-trivial, that's for sure.
All right. So as you mentioned, for the Moderna vaccine, the lipid is really important key component. So when you finalize the lipid, I think you may have screened so many lipids from your library. So when you screened the lipid library, how did you select the final one? Can you share with us what is your selection criteria for finalizing the best one, the right one?
Yeah, well, let me tell you how we've done things at MIT. You know, which is probably easier, and we've published, Moderna's published ways that they do it, but you know, you can take different lipids and look at expression. First, you could do luciferase assays in vitro, and then you could do like factor 7 assays in vivo. You know, that gives you pretty good readouts. That's certainly a good way to start. But then, of course, you ultimately have to use animal models, and look at expression of whatever messenger RNA you're looking for. And then you also have to look for toxicity. And, so ultimately it's going to be by doing things in small animals and then large animal models.
All right. So actually, you use your cationyl lipid is ionizable lipid, right? So do you think ionizable lipid, as compared to conventional cationyl lipid, what do you think is the unique advantage of ionizable lipid? Do you think it's really essential?
Well, I think it's certainly been shown to be promising, and I think that people are using it. I think getting the right complexation, that will, break up at the right pH is certainly important. But I think when I really look at the evolution of particle design, going back to even, like I say, what we did in 1974, I have no doubt that as time progresses, people will continue to make better and better nanoparticles and better strategies. Exactly what they'll do, it's impossible to say. I mean, we certainly work on different things ourselves, and as do many others, but I think there's been a consistent evolution of doing things better and better from, like I said at the beginning of our talk, people didn't even think it was possible to make nanoparticle or microparticle drug delivery systems. Now it's gotten kind of commonplace, but it's great that the various people around the world, including in Korea, are making better and better ones. And I believe and I hope that will continue.
All right. So actually, you are really, I mean, you are expert of many things, but you developed the PLGA, the microparticle part. So there are many papers about the cationic polymers for gene delivery. However, it turned out that cationic lipid actually won. So what do you think is the advantage of a cationic lipid over cationic polymers? I mean, in your opinion, why cationic lipid really predominate in terms of the approval game?
Well, I think it's still, by the way, different companies are looking at both. I mean, and both have advantages and disadvantages, but I think the lipids are taken up by the cells. And that, I think, has been a significant advantage, that that the cells take up the lipid particles because of, their lipophilic nature. But my belief is that polymers will be useful too. And that gets to what and I think they have. I mean, but I think that we're looking at a point in time, and I think that my feeling with all of the great work going on by so many people around the world is that I think we'll continue to see advances, whether they'll be polymers or lipids or micelles or things that we haven't even imagined. I'm sure there'll be more advances in the years to come.
All right. And also in Moderna vaccine, you modified mRNA with pseudouridine. So that kind of modification is kind of helpful in alleviating some kind of side effect. But I think Moderna also, as far as I know, Moderna has a lot of mRNA modification technology. So what do you think is the promising future direction of mRNA modification? I mean, unless it is not confidential, can you share with us about what would be the future mRNA if it is not pseudouridine modified?
Well, I think there's a lot of different things that could be explored. We have a fantastic chief scientific officer at Moderna, Melissa Moore. And I mean, I probably can't say exactly what everybody's doing, but I can say that, she's a superb molecular biologist, and she and her team have been, certainly looking at a number of different modifications to, depending on the application and use.
So some people have some concerns that that kind of mRNA vaccine, when they are taken up by the cell, the mRNA can interact with some microRNA or some other small RNA fragments in the cell. And do you think that kind of their concerns are really feasible to consider? I think that kind of interaction of mRNA with small RNA and endogenous mRNA in the cell, do you think it is really feasible scenario?
I don't think that we've seen any significant issues, safety issues or other issues with that. So I don't want to say that anything's possible, but I would say the safety of these systems have been extremely good.
All right, okay. And I think it's really historically rapid that the Moderna really got approval of the COVID vaccine. But even though in such a short period, I think Moderna have faced some kind of regulatory hurdles. So during from the your concept to the real product, what is the major regulatory hurdle, if you remember?
Well, I think actually the only one that's really come up is recently for certain people of a certain age and possible concerns about heart problems. But I don't even know if that's a hurdle. I mean, I think the FDA just wanted more time to look at that, and they're incredible. The FDA is, they're working unbelievably hard on, approving our vaccine and other vaccines for different things. So I actually don't think it'll be a significant hurdle. And time-wise, just in my own experience, I was once chair of the FDA Science Board. I mean, there really have been minimal hurdles for Moderna. I mean, I've been in cases with other things we've developed where there have been 3 or 4 year hurdles, and I mean, this might be a couple months. I mean, it's and it's only for a small population of people, so I think it's quite minor. But I think it's good that the FDA you want the FDA to be cautious, and I think that it's always this point of first do no harm.
All right. So actually now the Moderna vaccine, it should be transported by the cold chain. So do you think that kind of stability, the freezing in very low temperature, is it due to very short development period, or it is really due to some stability issue of lipid nanoparticle? What do you think about the stability issue during storage?
Yeah, well, the Moderna nanoparticles, of course, are quite a bit more stable than the Pfizer-BioNTech nanoparticles. But ultimately, you have to think about the fact that lipids are usually in what's called a metastable state. So, so that means that, they can change their character over time. I think at Moderna, they optimized it tremendously so that you can actually store them in a refrigerator for quite a long time. But ultimately, if you do things for many, many months at a temperature like that, then it could go bad. I mean, that's what happens with a metastable state. When you were asking before about polymers and lipids, that's an advantage. Polymers are not going to be in a metastable state. But on the other hand, the cells take up the lipids better. So, that's what's being used. But maybe, we and others have also been looking at sometimes hybrid systems. And so I think there's all kinds of possibilities for the future.
All right, so you think stability is still an issue to be improved, right? All right.
I think there's a number of issues, sure, but I mean, and by the way, this is just for a COVID vaccine. There's other things that, it depends on the problem people are trying to solve. But that being said, I think, there's no therapy out there that can do what messenger RNA did. You can't trade. I mean, all these things could be an issue, but the efficacy and the safety of these vaccines have been, remarkable, as well as the speed by which they can be made.
So, for example, in the siRNA, the product, do you think the siRNA lipid nanoparticle also need to be stored at such a low temperature, or siRNA lipid nanoparticle has less stability problem?
Well, if there's two issues, right, on any stability. One is the messenger RNA, which has its own stability issues, and the other is the lipid nanoparticle. I think you have to look at both of those. siRNA is inherently, in my opinion, it's a smaller molecule than messenger RNA, so it's generally going to be more stable. So I think you have to look at everything when you make an assessment.
I see.
So you'd have more flexibility with siRNA than you would with messenger RNA.
I see. So that is actually in part contributed by the low stability of mRNA as compared to siRNA.
I think that's fair.
All right. Okay. All right. So actually, nanomedicine has been widely studied in the field of cancer therapy, but it turns out that now the infectious disease is the more influential field. So after this, the mRNA vaccine, what do you think is the most promising field that the mRNA delivery technology can be applied to? What is your forecast?
Yeah, well, I think you just want to look at the Moderna pipeline, which is, I think, a pretty good example of this, and maybe the BioNTech pipeline. But you know what Moderna is doing, and we're in clinical trials for, I think, 8 or 9 other vaccines. You know, like flu vaccine, RSV. I mean, then we're in clinical trials for cancer vaccines. We're in clinical trials for new cancer treatments, clinical trials for heart disease, clinical trials for rare diseases, clinical trials for cystic fibrosis. So I think that there's a lot of different things that that I think you'll see in the future.
So like the virus infectious disease, that kind of the virus itself is really, I mean, it's clear. However, in the cancer vaccine, do you think cancer is really heterogeneous? So do you think cancer vaccine is really promising, like the mRNA infectious vaccine? What do you think is the future of cancer mRNA vaccine?
Well, I'm hopeful that that will be a whole new strategy to treat cancer. Of course, you know, when you're doing infectious diseases, you know, that's what I'll call more a prophylactic vaccine, right? You're giving people something before they get the disease, so that if the COVID or whatever flu comes, you know, your body's set up to attack it. In the case of cancer vaccines, it's what's called the therapeutic vaccine. So you're taking and they're even coming up with personalized vaccines, that you look at the patient's cancer and then you design, based on that cancer, maybe 24 or 36 messenger RNAs that will attack that cancer and hopefully destroy it. So it's I think, a very exciting idea. And, you know, there's been some promising initial results, but, you know, it's still early.
All right. So you are the giant in DDS field. So as a giant in this DDS field, what do you think is the next generation DDS system? For example, in 2030, what kind of new DDS system do you expect? Do you imagine?
Well, I think there are a lot, right? I mean, I'll just give you a few. I mean, I think we'll see better and better delivery systems for genetic therapies, including things like gene editing. I think we'll see, like I mentioned in my talk, I mean, possibly we'll see oral delivery systems for complex molecules. I also think there's lots of other barriers that we want to come up with better ways to get through, right? Like the brain, the skin, you know, I mean, the eye, the vagina. I think there's all kinds of things that you'll see, and people are working on all over the world, you know, on many of these. So I think you'll see a lot of different exciting things.
And in your talk, you just mentioned about the brain nanochip. Can you tell us more about the concept of brain nanochip? What is it?
Well, basically, it's work that Li Wei Sai and ourselves have been doing. We have an NIH grant. We've had a wonderful postdoc, Alice Stanton from Stanford, and you know, basically, we're trying to mimic what the brain is doing by growing the cells and the different cell types, you know, and creating a vascular structure to mimic the blood-brain barrier. So, but then the idea is, if you had that, you know, that, and you validate that, which is what we're doing, you know, that might be able to be used for tremendously accelerating drug testing for, you know, the blood-brain barrier delivery.
I see. Now I will ask a more general question. I think you have a great record of making many startup companies. So can you give some kind of advices or suggestions for some professors who are planning to or about to initiate some startup company? Any suggestions?
Yeah, sure. Well, I would say first you need very good investors. Secondly, you need very good, meaning by that they're not just give you money, but that they understand that medicine takes a long time, so they have to be patient and, you know, to get the reward. And they'll stick with the company in good times and bad. That's a good investor. Second thing is good business people. Third thing is good intellectual property, good patents. Obviously, the fourth thing, and I'm not putting these in any order, is great scientists.
All right. So the Boston area is really unique because there are so many bio ventures and there are really active collaborations between academia and bio venture. So what do you think is the actually that kind of historic background for making Boston such a unique environment? Do you think some government, the state policy really has done something to make Boston such a mecca of bio venture?
I don't. I mean, my personal feeling, and of course I'm prejudiced, Cambridge and Boston has two of the greatest universities in the world, Harvard and MIT. And, you know, in some rankings, they're ranked 1 and 2. And MIT, I think, actually has an engineering culture and is almost always ranked #1. I think just the proximity to those universities with the tremendous students that come out of there and have this kind of entrepreneurial attitude and are great scientists. That was one of the things I just said. I think that's unique. I don't think there's any place its size in the world like that has Cambridge, and here you have Harvard and MIT, you know, 2 miles away. So I personally think that's the #1 thing. And with that, everything else comes. I mean, venture capital comes, lawyer comes, you know, but I think it's really having those two great universities.
All right. All right, so great scientists and great academic environment, actually they are attractive, everything, yeah, all right. So, and many people are really curious about your excellent, I mean, never daunting motivation for driving the research. So what is your motivation for doing science?
Well, it's twofold. One is I love science. I mean, I love doing, you know, I'm curious. I love seeing experiments, you know, that can teach us something. I also love working with my students and postdocs. And I also want to see science do good for the world, make the world a better place. So it's all, you know, all those things.
Although you are now the world giant, you already mentioned that in your talk, you got 9 grants rejected in your early days, right? So when you have that kind of difficult time, how did you overcome that kind of the difficulty? What was your strategy?
I just kept trying. I wish I could tell you I had some magic formula. It was very discouraging, but you know, you have 2 choices. You can give up or you can keep trying. So I did the latter.
Just keep trying.
It didn't make me happy.
All right. All right, so almost the time is up, but my final question is that now there are many young scientists who are just attending this session. So do you have any message to the young scientists?
Well, when I give commencement speeches and things like that, what I usually tell people is to dream big, you know, dreams that hopefully can change the world and make it a better place. I think if you do that, a lot of times, just like I did, you'll run into people criticizing you and telling you it won't work. And I just say, don't give up. I mean, keep trying for your dreams.
All right, thank you so much. So keep trying and dream big. The take-home messages. Thank you so much, Dr. Langer, for great and inspiring talks. Bye-bye. Have a nice day.
Bye-bye.
Bye.
You too. Thank you so much. Bye-bye.
Thank you. I really enjoyed my honor.
Me too. Thank you.
Bye.
Bye-bye. Bye.
늦은 시간까지 같이 참여해 주신 여러분들께 감사의 말씀 전합니다. 예, 저희 행사는 여기에서 종료하도록 하겠습니다. 감사합니다.